Today I'd like to talk a little bit about 5-HT1A Autoreceptor Desensitization and a theory that I have for it. So in order to begin, we have to step back and look at the nervous system as a whole. The nervous system is broken into two broad categories, which would be the central nervous system, or the CNS, and the peripheral nervous system, or the PNS. We're going to be focused mainly on the CNS, which is the spinal cord in the brain, up here. But you also have to understand that the peripheral nervous system, which is mostly nerves, it covers the whole body. And so what you call a motor nerve or an efferent nerve is a signal that's coming from up here, the central nervous system, and going down. So motor would be, you're standing and you're moving your leg. So that'd be a signal down. And then afferent or sensory would be, starting from a nerve and going up to your central nervous system. And so an important thing to understand about neurons is that neurons are cells too. And so just like any other cell, they have parts that every other cell has too. So they share that. And so some of the important parts that we'll have to focus on would be the nucleus, which is the area of the cell that contains the genetic material, the DNA and the chromosomes. And this is where the cell, it's where all of the genetic stuff is going on. The mitochondria are what produce energy to fuel cellular activities. So the mitochondria are necessary for giving energy to the cell to do what it needs to do. And the synapse is not labeled in this diagram I have here, but it's actually the area between neurons where chemical communication actually takes place. This is where neurotransmitters are released to send chemical signals throughout the brain. And so that would be between the next two components of neurons that are specific to neurons, and that would be axons, which are used to send electrical information. So that's the presynaptic side of a synapse, and then dendrites, which are the part of the neuron that receives information, making them the postsynaptic side of the synapse. And they can receive, and they do receive, from many different neurons at once. So there's two basic types of neurotransmitter receptors in the brain. And the first type is metabotropic receptors, and the second is ionotropic receptors. Metabotropic receptors act on ion channels through a secondary messenger. So if you look up here, this is an example of a G protein coupled receptor, which is a type of metabotropic receptor. You see here, this is the neurotransmitter, the green dot, and it's activating this receptor. And when you look over at the ionotropic receptor, you see that the neurotransmitter is directly opening the channel that's letting through these ions, which is sending the signal to the next cell. But in the metabotropic receptor, or the G protein coupled receptors, there's this secondary messenger G protein that is sending a message to the ion gate, the ion channel, that is then letting the ions through. So there's a pathway that doesn't just involve the receptor. There's more between the two of them than just when you have the ionotropic receptor, where you have just the neurotransmitters opening the gate. And additionally, receptors can either be a heteroreceptor, which influences how the cell manufactures and releases neurotransmitters different from the agent that stimulated it. And then the other kind of receptor, it would be an autoreceptor, and that's located on the presynaptic membrane. And it regulates the levels of neurotransmitter released from the cell it sits on. It senses, in this case, it senses how much serotonin is currently in the synapse. And so it tries to regulate those levels, which I'll talk about in a few minutes, but it tries to regulate those levels based on sensing how much is in the synapse at one time. And serotonin is found in the central nervous system. It's also actually found in the peripheral nervous system too, but in the central nervous system, it's very, very, very important for mood, sex, appetite, sleep, memory, and learning. And it's the target for SSRI antidepressants. This SERT, or this 5-HTT, is the transporter that uptakes and terminates serotonin signaling. That would be on the presynaptic side of the synapse. The Ralph nuclei are 9 midbrain nuclei that release most of the 5-HT in the central nervous system. 5-HT is serotonin. It's a different name for the same thing. And right here I have the serotonin molecule. And I'll talk about this more in a little bit too, but there are 3 major families of serotonin receptors. But there are 14 different types. So it's not like you have one type of serotonin receptor in the brain. There's a lot of them, and they're actually broken into categories of how they work, and they're actually very, very different. So I talked a little bit about the Ralph nuclei, which are right here in the midbrain. And so this is actually the starting point of the serotonergic activity in the brain. And so from this point, there are projections that go, to the frontal cortex, to the hippocampus, the nucleus accumbens. They're all across the brain, but they all start from that Ralph nuclei. This is another diagram that's helpful in kind of explaining how serotonin works and how SSRIs impact it. So here you see the SSRI, which is interfering with this SERT, or 5-HT, which is clearing the serotonin out of the synapse. And so once you do that, the idea is that the 5-HT will build up in here. And the monoamine theory of depression would be that you don't have enough serotonin, you're not having enough serotonin signaling, and so by increasing the amount of serotonin here, you're alleviating depression. So that brings us to the 5-HT1A autoreceptor, which is 5-HT1A is one of the 14 types of serotonin receptors in the brain, and the autoreceptor is a certain type of 5-HT1A receptor that is on the presynaptic side of the neuron. Kind of. It gets a little bit more tricky than that because you have the somatodendritic autoreceptors and the presynaptic autoreceptors. So the brain is checking itself on its serotonin release in two spots. And this diagram is good, but it doesn't say here is that, yes, there are serotonin 5-HT1D and 1B receptors, but there are also these 5-HT1A autoreceptors here. Then this is the presynaptic. This on the left here would be the dorsal Ralph nuclei, or any of the Ralph nuclei actually. And this would be where the neuron starts. And then this is a long distance, and this is the projection area. So you have these autoreceptors in an area like the hippocampus or the nucleus accumbens, but you also have them back in the Ralph nuclei. And that's where the somatodendritic autoreceptors are. And what they do is they're basically checking this signaling before it even leaves. And so if this is releasing serotonin back here, and then the autoreceptor is noticing that, it's going to be sending less signals to these sites across the brain. But even once it gets all the way here, there's still autoreceptors checking over here too. So when more serotonin is found in the synapse of the Ralph nuclei, binding of autoreceptors inhibits the release of 5-HT and the projections of the Ralph nuclei neurons. So that's like I was just saying, you have more 5-HT1A autoreceptor binding back here. It's inhibiting the cell from sending signals this way, and then as a result, there's less serotonin released. A good question you might have, and what is actually a problem with this, is that wouldn't you think that increasing the serotonin here by blocking the 5-HT or SERT transporter would lead to more serotonin, which would then lead to the somatodendritic and the presynaptic autoreceptors decreasing the amount of serotonin released. So wouldn't you expect that serotonin wouldn't actually be raised at all? And that's a great point, and that's actually what happens for the first approximately 4 to 5 weeks of antidepressant treatment, is where the serotonin levels don't actually increase that much because of the autoreceptors working against it, against SSRI, in the sense that it's negating the effect of the SSRI by just sending less, releasing less serotonin. And so, yes, the serotonin isn't being reuptake as much, but at the same time, not as much as being released. But then eventually, this leads to the desensitization of the 5-HT1A autoreceptor, which is when you have the serotonin levels actually increase substantially. So what's causing this desensitization? And this is an important question for the treatment of depression and for the creation of safer drugs with less side effects. If you can get very, very specific, if you can get to the cause of this desensitization, you can get around the whole messing with 5-HT reuptaking serotonin, and it'd be much faster too. Multiple studies have suggested that SSRIs induce desensitization of 5-HT1A autoreceptors, and that this occurs downstream of the 5-HT1A autoreceptor itself. So earlier I was talking about metabotropic receptors, and this 5-HT1A autoreceptor is a metabotropic receptor, which means that it has these G protein signaling molecules, and that there's the receptor, which is very different and separated by this distance from the ion channel, which is actually making change in the neuron. And so the studies have suggested that the desensitization isn't occurring here. So, for example, there's not something on this receptor that's blocking it. The receptor's not deformed. They're not being formed differently. That the change is actually happening downstream. And how they know that, I'll get into in a little bit. So, in the dorsal rough nucleus, GABA, which is another neurotransmitter, so GABA B receptors use the same intercellular G protein pathways as 5-HT1A autoreceptors. And so chronic treatment with SSRI fluoxamine reduces both 5-HT1A autoreceptor and GABA B receptor mediated GARK currents, or GERK currents. And so this suggests that the sensitization of the 5-HT1A autoreceptor by SSRI treatment occurs downstream of the receptor, because SSRIs work on serotonin, not GABA, and so one would think that if it's occurring, and it's known that they use this same pathway, so if it's occurring in both of those pathways, it would just make sense that there's nothing changing here in the 5-HT1A autoreceptor desensitization, but it's actually occurring here downstream of the receptor. So that's when they say downstream of the receptor, that's what they're talking about. So this is the receptor right here, and then after this is activated, these G proteins then leave and activate the ion channel. So it's after the receptor, but it's before the ion channel. And so people might be kind of confused at what I'm talking about when I say an action potential, or that something's or that autoreceptor is inhibiting serotonin from being released. And in order to understand that, I could spend hours describing the action potential, but it wouldn't. I can break it down a little bit more than that and make it a little bit quicker. And so how I would do that is I'd say, this is the outside on this diagram. This is the outside of a neuron, and down here is the inside of a neuron. And so constantly, it's happening now in your body, happens at all times, regardless of whether you're on an SSRI or not. Your cell is pumping out this sodium, this Na+, and it's pumping in this potassium, or K+. And what that does is that creates a gradient. And so how cells communicate with one another is electrical, but it's electrical only because they have these gradients of these different ions. And so because of this this pump, you have a high concentration of sodium on the outside of the neuron, and you have a high concentration of potassium on the inside of the neuron. And this is actually how these channels change how much serotonin these cells are releasing. And so when you look at the voltage of a neuron, it's going to always start at its resting state in the negative. It's going to always have a negative initial voltage. And it fires when it reaches this threshold, which is more positive than the voltage. And so the inside of a cell is going to be negative, which is kind of counterintuitive, because what I'm going to say is that it involves these positive potassium ions. How can these positive potassium ions make a cell more negative or less likely to fire? And that's by leaving. So once you get this high concentration of potassium on the inside of the cell, they want to leave. They don't want to be next to each other anymore. They want to get out. There's a strong electrochemical gradient. So there's this desire to go out of the cell. So when these GERK channels are open, the potassium is flowing out. And so when you have this, it's already a negative cell. And so when you have this positive ion rushing out, it just gets more negative. And so you don't really see it on this diagram, but imagine here that this is the cell at resting state, and it needs this much in order to fire. But if you let all this potassium out by activating these GERK channels, you're making it more negative, so it's just harder to fire. And when it's firing, it's when it's releasing serotonin. So these opening these GERK channels is making the neuron less likely to release serotonin, which overall leads to a decreased release of serotonin when the 5-HT1A autoreceptor has been activated. So what does this mean? So it means that opened GERK channels, like I said, in the Ralph nuclei and the dorsal Ralph nuclei, inhibit the release of serotonin. So when these are open, there's going to be a less substantial release of serotonin. And so when 5-HT1A autoreceptors are desensitized, there's less GERK channels opening, and that means that serotonin is released more freely. And that's when you get the increased release of serotonin after 4 or 5 weeks of an SSRI antidepressant. And so the result of this is more 5-HT neurotransmission and the release of 5-HT to projection sites. So I talked about it a little bit before. So there are 7 5-HT receptor families, and that's 5-HT1 through 5-HT7. And they can be divided, like I said a little bit earlier, into 3 major groups depending on which G protein signaling pathway they activate. And those 3 different types would be, first of all, so you can see right up here, the 5-HT1 receptor family. And so those couple mainly to GIO G proteins. And then 5-HT4 through 7, those are through GS pathway, so the GS G protein pathway. And then 5-HT2 receptors work through the GQ G protein pathway. And then the one that I didn't mention is 5-HT3 receptors, which are ionotropic. And this is really interesting because you can just see within the serotonin receptors there's so much variability, and some of the receptors are so intrinsically different. It's kind of an interesting fact, not really what we're going to be focused on here. We're going to be focused on the 5-HT1 receptors, so those would be GIO pathway for the G protein signaling pathway, which would be right here. So my theory is a G protein model of 5-HT1A autoreceptor desensitization that occurs downstream of the receptor, and that explains the autoreceptor desensitization. And more specifically, it's going to go into the regulation of G proteins and how regulators of G proteins are what I believe are causing the desensitization of 5-HT1A autoreceptors with SSRI treatment. The clinical implications of this are very important in creating new antidepressant treatments that work quickly and effectively raise serotonin levels in patients who cannot wait for treatments to work, or patients who are treatment resistant to current SSRI medications. Alpha subunits. This 35S GTP S, it's an assay used because it binds to alpha subunits. And alpha subunits are part of this G protein signaling complex here. So when this metabotropic G protein coupled receptor, which would for us be 5-HT1A autoreceptor, is activated by the signaling molecule, which in our case is serotonin, this complex is released. And so what this assay does is it binds to the alpha subunit, which is right here. And so you'd assume that if this were present, you'd have a lot of binding because you'd have a lot of alpha subunits. Little surprise, the dorsal Ralph nuclei and the hippocampus show high levels of 5-HT1A stimulated binding. And then that suggests that these regions are populated with 5-HT1A receptors using G protein signaling, which we already knew, but it's just kind of backing up what we had expected already. So this increase is after they're giving 5-HT1A agonists, or basically simplified, what that would be is a fake serotonin. So what's that suggesting is that if you're increasing the serotonin acutely or for a very short amount of time, you're getting more of these G proteins being activated. But this increase in the agonist, of this 35S GTP gamma S binding is totally abolished in the dorsal nucleus after 14 days of treatment with the SSRI fluoxetine, which I talked about earlier. It's Prozac. It's the first SSRI, the most common SSRI. And this suggests that desensitization of autoreceptors is happening here. And why would that be? Well, you're still adding the agonist, which is the signaling molecule, right? But you're not getting these alpha subunits being bound to. So basically what's happening is what's happening when you have the desensitization. So you're having the serotonin, or in this case the agonist, but downstream of that nothing's happening. And so this is just further proof that it's happening downstream in the G protein area instead of the receptor itself. So after these 14 days of treatment, this assay shows more binding in the hippocampus, which is counterintuitive until you think about it. If we go back to this slide, think about it. You have over here, you have the autoreceptor working less well. It's desensitized. So what's happening is there's less binding here. The binding increase is abolished. But as a result of that, there's more serotonin being released in an area like the hippocampus. And because of that, there's more binding on the postsynaptic side of the membrane in the hippocampus. And the 5-HT1A receptors still use this same pathway, even though they're the heteroreceptors on the postsynaptic side and they're not autoreceptors. So you would expect with more serotonin and more serotonergic activity in the hippocampus that there would be this increase in the G protein, there'd be this increase in the G protein activity of the alpha subunits here, and you also would expect a decrease here. So it's just showing us what we already would expect. And some more proof that this occurs downstream is that desensitization of 5-HT1A autoreceptors can be seen without changes in their density. And this has actually been found in multiple studies, and it's a little bit of a controversial topic, and something that when I first started looking at this debate, I was very much of the opinion that what's causing this desensitization is a decrease in the transcription of these 5-HT1A autoreceptors. But the more I've looked into it, I've seen these studies which are counterexamples to that, and evidence of this assay right here, and back here where this study right here, this 1987 by Innes, this study where they have the GABA receptors, both pathways are being desensitized. So that evidence together suggests to me that this is occurring downstream. And so the next question obviously becomes, what is causing this decrease in these G protein subunits after long-term treatment with an SSRI antidepressant? So, if you break down the G protein alpha subunit family, and we have to understand again that an alpha subunit is just part of this complex that is released, and as you can see here, it is interacting with ion channels. But if you see that there's a reduction in the level of these G protein alpha subunits, GI and GO, which are different types of G protein subunits. And if you go back here, we see that 5-HT receptors signal via GI and GO alpha subunits. And so you see a reduction of these levels in the midbrain, which is where the Ralph nuclei are, after chronic SSRI treatment. And so it is known that GO proteins are coupled with somatodendritic 5-HT1A autoreceptors, or receptors, but because it's in the midbrain, we know that these are autoreceptors. And so a decrease in their levels might be related to the desensitization that's observed in the binding assays showing 5-HT1A autoreceptor desensitization with less G protein signaling. And additionally, there's an increased level of GI1 and GI2, which are even more broken down components of this G protein family. And in the hippocampus, have been increased in the hippocampus following chronic administration of an SSRI antidepressant. And so this gives more support to the binding assays with the 35S GTP gamma S assays, where we see more of this G protein signaling in the hippocampus after the desensitization has occurred, and less of it in the midbrain or the Ralph nuclei. And so these GO and GI2 subunits in the midbrain are significantly reduced after only 3 days. And this suggests that the 5-HT receptors and the presynaptic and postsynaptic sides couple to different G protein subunits. Because over here we're seeing this increase, and over here we're seeing this decrease. So different parts of the serotonin system are using these subunits differently. So the theory with this is that after the dorsal Ralph nuclei or the Ralph nuclei, 5-HT1A autoreceptors are desensitized by fluoxetine within this 3-day period. And so these data also suggest that GO and GI1 subunits are used by autoreceptors, while heteroreceptors are more likely using these GI1 and GI3 subunits in the hippocampal projections. And this would support other findings in the literature that say that somatodendritic 5-HT1A autoreceptors in the Ralph nuclei are coupled to GO proteins and lead to an increase in opening of these potassium channels.